Okay, Assalamualaikum and a very good afternoon to all. Okay, so let's see our next new chapter, okay, about the light detectors. Right, so we'll see what are the subtopics that we will cover here. So we have to learn about the visual observations, okay, about the photograph, photoelectric detectors, photomultipliers, photodiodes, image intensifier. Okay, also the CCD, you have learned about the CCD before. So this is just some of the revision and additional information about the CCD and also the operation. Okay, and the interesting parts also about the video astronomy, how to take the video, proper video, and what have to take into account. And thermal detectors, comparison of the detectors, okay, color detectors, and also about the active pixel sensor. Okay, so for the first visual observation, we see the basis of before we go into the visual observation. So we must take consideration into our eyes structure. Okay, so this is the basic of the eye structure. So you know the visual observation is commonly referred to the process of gathering the information and the data through the sense of sight. Okay, so this method involves actually the direct observation, okay, and also the interpretation of visual and stimuli to understand and analyze a particular subject or any phenomena. Okay, so for thousands of years, people recorded what they saw in the sky. Okay, and on the rock walls, clear tablets, ivory, and paper. So until 19th century, human eyes was only available light detector. Okay, and the eye is essentially a spherical object that maintains its form by the sclera. Okay, so you can see from here, we have this the optic nerve. Okay, and the iris, lens, cornea, the main part, and also the retina to protect your eyes. Okay, so the front center portion of the sclera is the transparent cornea here, and also the crystalline lens. Okay, and it's separated by the liquid called as aqueous humor here. Okay, so the eyeball is filled with jelly-like vitreous humor, and which also helps to maintain its shape. So these four items determine the optical characteristic of our eyes. Okay, so the structure of eyes is such that rays that may enter the eye from direction nearly 180 degrees. Okay, for retina, is the inside lining of the eyeball filled with the photodetectors. So in order to focus the object on retina, the eye will change the focal length of lens, okay, and use the muscle surrounding the lens. So this process called as accommodation. So as we become older, the muscle that adjusts the lens will become less effective, and we find it increasingly difficult to focus, okay, on the nearby object. So we compensate first by holding object farther from the eye, and then by wearing the reading glasses. Okay, this process reached to the adaptation when our eyes possesses remarkable ability to adjust for difference in the level of the brightness, okay, or the illumination. So this is the adaptation. So part of this ability will result from the presence of iris diaphragm that located in front of our lens. So it's kind of muscle that can expand and contract within the opening range of 2 to 8 millimeter, depending on the total illumination or the brightness. So the opening in the iris will define the hole known as the pupil of the eye. Okay. Okay, so the color of the iris will determine the color of the person's eyes, okay? And the diameter of the iris, however, only offers a ratio of 1 to 16, okay? Hardly enough to account for the great adaptive power of the eye. Okay, the surface of the retina is covered with rods and cones. Okay, so both of these are sensitive, okay, to light differently. Okay, for rods, it responds to faint light more effectively than the cones, and the cones serve better in the bright light. Okay. Okay, so let's see the daylight vision, nighttime vision, and also the light range. Okay, for a better overview, I show you this comparison on 3 different vision. Okay, so we have this photopic, mesopic, and scopic. So you can see the differences in our vision when we have this photopic. Okay, it occurs the day or in bright light and increase color perception and also the sharpness. Okay, so this photopic it relies on both rod and cone photoreceptors. Okay, but for mesopic vision, okay, it occurs during twilight. So mixed color perception and mostly during nighttime lighting, okay, is a mesopic vision, and also rules on both rod and cone. And then we have this coptic occurs at night or in dim light, okay, and it's decreased the visual causes the loss of the color perception. Okay, so it requires use of peripheral vision and heavily relies on rod photoreceptors. So these are the 3 type of visions that's stated here. Okay. So this explains why when we wish to see a very faint object in the telescope, so we often have to look slightly to one side of it, and we find it hard to distinguish its color. So there is another small region lying just outward of the optical axis here of the eye, where only cones are found and in the greatest concentration. Okay, so these are called as the fovea centralis. Okay, so this one, this we call as the case lah. Okay, and also the spacing of the cones determines the acuity of vision, and also the smallest detectable angle of the eyes, about 1 degree. So this can be seen within the region of the fovea with diameter of less than 1 degree. So this just big enough to contain the image of the full moon. And other factors that can produce is hyperacuity, means this greater than the normal acuteness of sight. So this include eye jitter and also mental processing. Okay. So if we don't have these both cones and rods, it is found as in the blind spot where the optic nerve leaves the eye. So that is the case where we have the blind spot vision. Okay, so when a photon, this is a fundamental part, okay, about the visual observation. So when a photon strike these both rod and cone, a measurable electric current is created, and is passed to the brain, which brain will recognize that stimulus and reach a particular part of the retina. So when the level of this brightness high, so the rods are not active and the cones alone are sending stimuli to the brain. So this sensitivity cones only 1% that of the roads. So when we go from well-lit room into nearly dark, okay, so from bright room to nearly dark one, so the cones are not able to detect the faint illumination, and for a time we will see nothing. And the eye will then switch from cone vision to rod vision, and will take a few minutes before large number of rods become active to achieve the maximum night vision. So it take time, okay, bila kita nak kita move from uh, bright vision, okay, bright area to nearly a dark area, and also from dark area to go move nearly to the bright area. So our eyes, our vision, okay, will take some time to correlate with the stimulus, okay, which respond from the brain through the stimulus and get into your eyes vision. Okay, so there are actually 3 types of cone distinguished by pigments within the cells and determine the wavelength region to which the cone will respond. So we have this wavelength with the peak intensity and sensitivity, okay, blue, green, and red, and our brain process signal from these 3 types of cones. So to give us a sense of color, and the useful range of sensitivity about this range, okay, and also the wavelength sensitivity of the rods that peaks at about a green 5,000 angstrom for the green wavelength. Okay, and the dark adapted eyes are rather insensitive to red light, which why astronomers will use red lights when observing. So the response time for rods and cones are this period of time, 300 for rods, cones is 75 millisecond respectively. Thus, rapid motion is the best detected in high illumination. Okay, so important note, much of what we describe as vision is in fact not optical but physiological, okay, psychological. So the eye is only a detector. What we see depends on considerably how the brain will interpret the optical stimuli. Therefore, this means that one's experience can help to locate faint objects such galaxy or planetary nebula. Okay, so with the help of this telescope advancement and short-term memory of image, astronomers were able to make accurate drawing of the celestial phenomena in the 19th century. So this is the example, okay, of the lunar craters Copernicus at Observatory of Romano in 1864. That is how they interpret the structure, okay, about the lunar crater, okay, a moon, permukaan bulan, and also this for Jupiter at Lick Observatory, okay, 1890. So this is how they come up with. Okay, so we proceed to the next part, okay, about the photography. Okay, so photography is the process to make the first astronomical photograph for cumbersome and impractical in most situations. such as by John Draper that record the image of the moon with newly invented this process. Okay, the first publishable available photographic process. Okay, and also in 1845 by these two Physio and Foucault imaged the sun in Paris. Okay, and also after that we have this dry emulsion. Okay, invented a cutting the exposure time to allow quick preparation and the production of the photographic plates. So visual observation must be completed at the telescope. However, photographic observation allowed the analysis to be done later and at the ease of the astronomer. Okay, so structure of photographic emulsion is such that photon can accumulate until an image form. So we have longer exposure time allow the recording of the object, which were too faint to be seen visually. So total exposure is product of intensity at the photographic emulsion and the duration of the observation. Thus, if the flux is half, the exposure can be kept constant by doubling the open shutter time. Okay, so when doing the photograph on the stars, so essentially just a small spot of graded diameters and also about the brightness densities. Okay. However, the density within the individual spot is not necessarily uniform, so both the diameter and density have to measure at the same time. And then the density of exposed and developed emulsion increases with the exposure time, and response of particular emulsion is described by the characteristic curve for that emulsion. Along the characteristic curve is found that at exposures within the certain range, density will increase linearly with the exposure. And therefore, a photocell can be used to determine the density of the developed emulsion, okay, by using current as indicator. So when we do the what we call the stellar observation, so it's referred to the relative brightness that can be determined from the density measurements. Okay, such as we have from Jiggle camera, Charles Fabry's design. Okay, so these two important device, okay, designed with their own role. Okay, for Jiggle camera, designed to move the plate continuously in random motion. But for Charles Fabry's design is for matching the position of stars, and it's placed in the optical system near the focal plane. So each one of form image of the objective of the plate. So the design is important to help for the stellar observation, okay, to make it to have more better quality of the stellar observation. Okay, so by using different exposure time to record image of same star, a magnitude scale can be made, and also doing some calibration. Okay, at last they will get the last major astronomical project is to use the plate photography was in second polar moon observatory sky survey. Okay, we have learned about the sky survey. So the key element in the photoelectric system is the photocell itself, so the detector. Okay, so there are three types of detector. Okay, we will learn about photoemissive cell, photoconductive and photodiode. So three main photo cell. So we can have some comparison between these three types. So for photo emissive, here use a photon that impinge on a light sensitive metallic surface, which cathode, and causing the emission of electrons drawn to anode. So the rate of flow electron from cathode to anode is a measure of the light, the flux. So photoemissive cell were widely used since 1930, okay, since the discovery of the photoelectric effect. Okay, and for early photoemissive detector is not very sensitive and but they had the broadest use among the astronomers, okay, for photoemissive, okay, photoemissive detector. But for photoconductive cell is the electrical resistance of materials such as lead sulfide, selenium, with the changes in the presence of light. So these cells were the first detector used in photoelectric astronomy for observation in the infrared range. So that it for photoconductive, and we have these photodiodes. It's a semiconductor device related to the transistor, which convert light into electric current. So that is the behavior of the photodiodes to convert the transistor the transistor, okay, from light to electric current. And this widespread application of photodiode in astronomy only happened after 1980. Okay, so this is the sum of description on the what we call the illustration of the how the photo multiplier photodiode works. Okay, so it's unlike photograph. Photoelectric is the lack of spatial resolution. Okay, while there is a linear density portion in the characteristic curve of photographic emission. Okay, isn't one for the photocathode. So this can be solved by using the Fabris lens design just now. So they place behind the filter in the photometer to ensure the image form is uniformly illuminated spot. So that is the configuration, the structure of this Fabris. In fact, the spot described here is just the exit pupil of the objective Fabry lens system. So the focal length of Fabry lens should be chosen such the image form is small enough to fit onto the detector. So if you see the plane mirror at the top, it will removable when in the position it allows the observation through the eyepiece here. Okay, and then we do have this diaphragm, small aperture at the focal plane, function to block out the sky surrounding the star. And the prism mounted behind this aperture allows the observer a magnified view of the target object through the second eyepiece here. So on night of poor seeing or when measuring a bright star, the edges of image may appear to be blocked by the aperture. So a slide containing a set of graduated apertures is required. Okay. So left to the filters amplifier. Okay, before we okay, I will continue photo multiplier in the next lecture. So I will finish this illustration. Okay, just now about the diaphragm, focal plane, prism, and then we go into the filters that are mounted on the slide and when combined with the spectral sensitivity of detector, So it allows the user to isolate a specific range of wavelength and complete the photoelectric system that have many forms but always incorporate at least one type of light sensitive detectors mentioned. So from the detector, electron will flow through the circuit to amplifier, okay, that enhance the very weak signal or a counter, and finally to a display with the computer lah, okay. So that is the description on the working of photoelectric detector, okay, based on this illustration. Okay, so I will stop at this slide. We continue in the next lecture. Okay, that's all for today. Thank you.